My name is Hiroyoshi Aoki, and I work for TOYO Corporation. In this presentation, I will discuss two of our products, the AGM Reagent Kit for microorganisms and the recently launched AGM Reagent Kit for animal cells. Earlier, we launched an agarose gel microcapsule, AGM, product for genome analysis and culture of bacterial single cells. An AGM is a hollow hydrogel microcapsule consisting of an agarose gel shell and a liquid alginate core. Since medium diffuses from the outside through a porous agarose gel shell, the AGM provides a micrometer-size culture vessel for embedded microorganisms, which is useful for high-throughput screening. Additionally, reagents of multiple displacement amplification, MDA, diffuse through an agarose gel shell while high molecular DNA products amplified using MDA are sustained in an AGM core. The AGM effectively performs in a picoliter scale reaction chamber to suppress amplification bias on the MDA, which causes serious problems during single-cell genome analysis. On the next slide, I will discuss our innovative product, the AGM kit for single-cell genome analysis. The single-cell genome analysis of environmental microorganisms is a promising technique for genomic identification of unknown microorganisms and SNP genotyping of microbial populations. According to conventional practices, microorganisms are isolated using FACS or a micromanipulator and amplified through microliter scale reactions with the MDA method before DNA sequencing. However, the procedure involves the long-standing challenge of amplification bias caused by a large amount of reaction mixture. Our AGM technology reduces reaction mixture in volume by gelling the agarose through two different emulsion processes according to the batch method and embedding microorganisms in the microcapsules. Notably, our kit supplies a special oil capable of stabilizing the without emulsions, which eliminates the need to use special equipment for AGMs. We amplified these genomic DNA sequences by the multiple displacement amplification method. Then, we isolated amplified DNA sequences using a micromanipulator and performed the genomic analysis. The chart shows a comparison of the MDA reaction in AGMs with the MDA reaction in microtubes, the conventional method, using different microbial samples. The comparison shows that the AGM technique, which achieved minimized reaction mixture volume, successfully suppressed MDA-associated amplification bias and increased genome coverage. Next, we tried to develop an AGM kit for animal cells to explore additional applications in single-cell genome analysis, cell transplantation, stem cell culturing, and tumor spheroid development. However, when our AGM kit for microorganisms was used to amplify animal cells without adapting the device, animal cells were killed, which was another challenge to be addressed. We optimized the reagent conditions and developed the kit for animal cells. The chart shows the manufacturing process for the AGMs containing the embedded animal cells. MCF7, a human breast carcinoma-derived cancer cell line, was used for testing. Cultured MCF7 was treated with trypsin and emulsified in alginic acid and oil to form an alginate core. You mix the core with agarose and gel the mixture once more in another emulsion to solubilize the core and create AGMs. The kit is compatible with common or inexpensive laboratory equipment and has the great financial advantage of minimized introduction cost. Cells were added to the liquid alginate and emulsified in oil using a digital stirrer. The distribution of core diameter was proportional to the stirrer speed. Indeed, approximately 70% of cores had a diameter of 50 to 150 micrometers at approximately 1,000 revolutions per minute. This figure shows the cell survival rates within the liquid alginate core for our conventional and new kits. Our conventional kit was associated with a problem: the cells were killed when liquid alginate was gelled. The new kit achieved a survival rate of more than 80%, including during the core solubilization process. The photos represent cell growth in actual cultures. Culturing grew cells fully within the cores and formed spherical cell colonies of spheroids. As we continuously cultured the cells burst the shells and leaked out. The finding means that we may obtain cells grown in AGMs if we simply continue culturing them. The photo shows another culturing exercise under a different condition. Cell growth was found to be slower within alginate gel beads, which are commonly used vehicles, than in AGMs because the alginate gels had higher resistance. In addition to MCF7, we cultured C3H10T half cells, which are mouse umbilical cord blood-derived cells in AGMs. They grew poorly. These cells have contact inhibition and are, therefore, unable to form scaffolds. Cells with contact inhibition may require a scaffold protein within the AGM to grow. The photos show viable and dead cells stained using live dead cell staining kits. Apparently MCF7 cells do not grow well in alginate gel, and C3H10T half cells also do not grow well in AGMs, but, In reality, they are not dead. Instead, they are alive within the media. In conclusion, we developed an AGM kit for animal cells. We tested the kit by culturing MCF7. The cells developed characteristic spherical colonies known as spheroids. Since the AGMs contain liquid medium within the shell, cells more easily grew in AGMs than in gel beads. Our AGM kit for animal cells will find many opportunities for use in future single-cell genome analysis, cancer research, stem cell research, cell transplantation, and more. Please visit our website for more information. Thank you for your attention.